Today, we're going to talk about something that's happening inside you right this second. It's one of the most fundamental processes in all of biology, the cell cycle. And we're going to look at a totally new, kind of revolutionary way scientists have figured out how to understand its incredibly complex hidden program.
But let me start with a question for you. When you think of the most complex machine on Earth, what comes to mind? Maybe a supercomputer? A particle accelerator? Well, what if I told you it was something else entirely?
Yeah, the answer might actually surprise you. It's microscopic. It is everywhere, and trillions of them are working inside you right now. It's the living cell. Just think about that. Trillions of these tiny machines all working together to make you, well, you.
And maybe the cell's single greatest trick, its greatest feat, is its ability to perfectly copy itself. This whole process, the cell cycle, it's literally the engine of life. But for decades, the instruction manual that tells it how to do this has been a complete mystery, a black box, until now.
So, to really appreciate just how big a breakthrough this is, we first have to get our heads around the scale of the problem scientists were facing.
The researchers behind this new model put it perfectly. They said that trying to understand what's going on inside a cell is just impeded by this staggering level of complexity. Imagine trying to map every single conversation, every whisper, every text message happening in a city like New York all at the same time. That's kind of the scale of the challenge we're talking about here.
See, the cell cycle involves thousands of genes and molecules, all tangled up in this vast, interconnected network. It's like trying to understand an orchestra with thousands of musicians, all playing different parts, through different movements, but there's no conductor and there's no sheet music to read. The old ways of modeling this stuff, they were just completely overwhelmed. They couldn't find the melody in all that noise.
So, to crack this problem, scientists needed a totally new approach, a new way of thinking. They had to stop looking at every single musician and instead start asking a different question. What song is the orchestra trying to play?
And this is where we get to the core of the breakthrough. It's called the cybernetic concept. And the big idea here is to stop treating the cell like just a bag of random parts, and instead to treat it like an optimal strategist. The model basically assumes the cell is smart, that it's actively trying to achieve specific goals at each stage of the cycle. Like, my goal right now is to grow, or my goal is to copy all my DNA.
This really shows you the difference in thinking, right? The old way was trying to map everything, every single side street, every little back alley in that city, and it was just too much information. This new cybernetic-inspired method, or CIM, is way smarter. It says, forget the local roads. Let's just find the main highways, the really essential pathways that get the cell where it needs to go. It's all about finding that critical signal in all the noise.
Okay, so how does this actually work in practice? Well, it's this really elegant two-step process that starts by, you guessed it, drafting a really rough preliminary map of all the possible connections.
So it starts with step 1. Draft this huge map of potential cause and effect links between genes using a pretty cool technique from information theory. Then comes step 2, and this is where the magic happens. The model applies that goal-oriented filter to find which of those links are actually essential.
So this first step, it uses a tool called Time-Delayed Mutual Information, or TDMI for short. And probably the easiest way to think about it is that the scientists are basically eavesdropping on the conversations happening between thousands of genes. By watching to see when one gene speaks or becomes active, and then measuring how long it takes for another gene to reply, TDMI can create this huge preliminary map of who might be talking to whom.
But that initial map is incredibly noisy, right? It's full of connections that might just be a coincidence. So now comes the really powerful second step, filtering that map to find the true master controls.
And I mean, just look at this number. That initial TDMI analysis came back with a massive, tangled web of 731 potential interactions, and that's just between the 86 key genes they were studying. This is that noisy, every street on the map version we were talking about.
But then they applied the CIM filter, the goal-oriented model. And from that chaos of 731 connections, the model boiled it all down to just 153 essential interactions. That's the power of this thing. It cut through more than 75% of the noise to reveal the critical pathways, the highways that are truly driving the cell cycle. And this isn't just about deleting data to make things look simpler.
By focusing only on those goal-oriented interactions, the researchers were left with a much clearer and, frankly, a much more meaningful map of how the cell is actually controlling its own replication.
So the big question, does this new map actually work? And the answer is a huge yes, but maybe not in the way you'd first think. Now, if you're looking at this table, you're probably thinking, hey, the predicted times don't perfectly match the real times. And you're right. But that's not the real story here. The real breakthrough is what the model found within those phases. For example, it correctly figured out that the S phase, that's when all the DNA gets copied, it isn't just one long event. It's actually made of 3 distinct substages. I mean, that is a level of detail that other models just completely miss. It proves this goal-oriented approach can see the underlying program, not just the final runtime.
And the discoveries just kept coming. The model didn't just rediscover what we already knew. No, it pointed out 14 potential gene interactions that had never been documented before. It found the key manager genes for each specific stage of the cycle. And, maybe most importantly, 90% of the connections it did find were already backed up by scientific literature. That tells you the model is both incredibly accurate and genuinely innovative.
Okay, so this brings us to what might be the most exciting part of this whole thing. Because the real breakthrough here isn't just a better map of the cell cycle. It's the map-making tool itself.
And this really is the biggest takeaway. The researchers didn't just create a new map for one process. They've created a whole new kind of map-making tool, a new lens, that could be used for pretty much any complex biological system.
I mean, just let your mind run with the possibilities here. We could use this goal-oriented approach to better understand how an embryo develops from a single cell, or to decode the progression of complex diseases like cancer, or even model how a stem cell decides to become a heart cell versus a brain cell. It gives us a brand new way to look at the fundamental programs of life itself.
And that leaves us with this final, really powerful question. If we can crack the code of something as complex as the cell cycle just by changing our perspective and asking, what is the goal? What other profound mysteries of life could we finally begin to decode?